Hi, it's Brian Kane from BPI TV. I'm here with Joy Concepcion, Global Marketing Manager with Gyros Protein Technologies. Joy, welcome.
Thank you.
Joy, running ELISAs for analytical testing is a major bottleneck. How does the Gyrolab platform help overcome that challenge?
So the Gyrolab platform is an analytical platform that automates and miniaturizes immunoassays at nanoliter volumes. Because ELISAs routinely require a lot of hands-on incubations and washes, it takes 3 to 4 hours to run. But our platform, with its miniaturization and automation, automates the entire workflow hands-free in under 50 minutes, while still producing 3 to 4 logs of dynamic range, and also allows you to run all the samples in parallel using only nanoliter volumes.
Gotcha. So certainly quality by design is a major initiative in bioprocess development and production. So how does the Gyrolab help?
So recently we launched the Gyrolab Expand platform. And what that does is that allows the analytical department to run multiple assays in parallel to support a lot of the different assays run for QBD initiatives. You can run different types of assays or different conditions. In addition, when you combine that with the automation, so whether you're running development or you're running QC testing or in-process sample analysis, you're always going to get consistent results that are not reliant upon manual workflows that really increase the variability of existing analysis. So when you take all of the manual handling out of the process, then a lot of your immunoassays are more consistent, more reliable, more robust, whether you're in development, in-process analysis, or production.
Great, thank you. Is the Gyrolab just an analytical platform, or are there other tools that you use for CMC biologics?
So while you can design or develop your own assays because it's an open platform, because in bioprocess development and CMC biologics, they really want plug-and-play solutions. They don't want to have to QC their own reagents. So what we've done is we've come up with a suite of analytical kits that are ready to use for cell line development and bioreactor monitoring for a human IgG titer, host cell protein analysis for CHO-K1 for downstream purification, and then also for leach protein A in downstream purification as well.
So are there any other applications that Gyrolab supports when it comes to immunoassays?
So we can also do characterization to measure affinity for in-solution kinetics. And then in addition to that, you can do a lot of yes-no binding. Because we're not just an endpoint assay, it uses an affinity flow-through format. So it's like running an ELISA on an HPLC column. So not only are you getting a rate that you can back calculate off a standard curve, we also give you a picture of where everything is binding on that column. And what that does is that gives you additional information to make better decisions, whether you're screening monoclonal therapeutics or clones, or you're doing assay development and you want more information for best pair analysis.
Gotcha. So in general, the Gyrolab helps. In a very complex process, the Gyrolab's objective is to simplify the process and provide end users with more information to make better decisions. Is that right?
Exactly. So our whole goal is to not just give you high performance immunoassays, but to increase your overall productivity and efficiency for bioprocess development, for downstream purification, and then production. So it's not just in the development of your CMC biologic, but it's also in the production of. It's been validated in GMP environment, and it's also used in antibody engineering, cell line development, upstream and downstream.
Great. Exciting times in the industry, very exciting times for Gyros. Joy, thank you very much for joining us today.
Thank you so much.